Good morning, everyone. First, I would like to thank Professor Chattopadhyay, Dr. Tony, and Dr. Soumitra for giving this opportunity and this platform, which is very important because this is a platform which can be addressed and can be we can talk of confocal microscopy to its core, the basic system in itself, and what are the samples which can be used to image, what should be the sample preparation types, and everything obviously should be guided by the objective of your studies. So this is a high-end system which will enhance your research work definitely. And so today I'll be covering onto the system potential, which is the ZEISS LSM 980 microscope. But before I start onto the system, how does a microscope work has to be like everybody should know. So a basic introduction to microscopy, as you can see, is a very primitive model of a microscope image you can see here. And a microscope is basically designed to produce magnified images, and which can be like later on documented and used for various studies. Now, the basic objective of the microscope is like to see or to magnify sub-resolved to human eyes. So objects or samples which are not resolved through human eyes, people have already started having curiosity to explore and understand those sub-resolved items. So as you can see here, based on the size, we can see how like different techniques have been used or has been used, which has been capitalized by researchers to understand and explore more and more unprecedented avenues and to find out more informations out of them. So microscopy basically works on three most important things. Obviously the magnification, we want to have a magnified image which is otherwise too small for human eyes to notice or to understand. But magnification alone won't help. There has to be a proper resolution to that. And obviously, if a resolved image, a magnified image, again, would have no meaning if the contrast or the details of the sample is not well described. So I see here a low magnification and a high magnification image, which is comparable here. So in this case, the sample is well magnified, but magnification alone won't help. So taking back to the basic schematic diagram of a compound light microscope, and as you can see here, just using two objectives and the eyepiece lenses, the entire magnification of a virtually inverted magnified image has been done. So keeping this basic schematic diagram at the back of your mind, you can calculate what is the magnification or to what extent you can magnify your sample. A magnified sample which is not resolved, yes, again, it has to be resolved. Otherwise, it has a lot of informations which you cannot decipher. And if we talk of resolution, then as like you can see here, the numerical aperture of the objective or basically the lens is very important. And how does, so you have this equation here, and based on this, the higher the numerical aperture, the better will be the resolution. So one must understand what is the numerical aperture and what type of lens or objectives they are using to acquire magnified images using the system. Now, this is something very basic equation, and as why it is important again and again, because the refractive index is another crucial property which is to be understood before selecting an objective. So if you are using, as you can see, this particular image, the ray diagram, so when light travels from a denser to a lighter medium, it always moves away from the norm. So keeping that in mind, if you see the light path, so when it travels from the cover glass and to the air, it always moves away from the normal. And because of this deviation, we are losing a lot of light which are to be collected. So this has to be corrected, and just it can be corrected so that using a immersion oil. Now this immersion oil has a refractive index which is very close to the refractive index of the glass, and thus matching the refractive index, the refractive index mismatch is low. So the light path deviation has been rectified, and then more of the light can be collected. So that is one thing where you are using higher objectives where the refractive index has to be, the mismatch has to be removed. Immersion oils are used which has a refractive index close to the refractive index of the glass. So as a common example which you have seen, like if you see a straw in a water, there's a deviation. So this has to be kept in mind, and then by using the appropriate immersion oil, or even you have to check the objectives. If it is a water dipping objective, then water has to be used and fill the gap between the objective and the glass. Then the last and the most important is obviously a contrast. So in case of a contrast, being important, because even if the sample is well resolved, the image is magnified. If there is no contrast, then the informations lying within that image makes no sense. So contrast, there are a lot of contrast techniques which has been used on and off, and with a special emphasis because we will be talking about the fluorescence dyes, because fluorescence dyes will be used to tag to specific organelles or the specific areas of your sample which you want to image. So if we talk like a sample is there which is tagged with your fluorescence dye, the fluorescence dye has a property which gets excited with a particular wavelength of light, and in return you get an emission signal. This emission signal is something which has to be tapped, and based on the bandwidth of this emission signal which we are tapping, we get the final image processed. So again, like if you do not forget the electromagnetic spectrum. So in this electromagnetic spectrum, the visible light or band which is visible to the human eye is ranging from roughly around 400 to 700 nanometer. So as you can see in the VIBGYOR, and the important thing which has to be kept in mind, as we increase the wavelength, the energy associated with the wavelength decreases. So they are like inversely proportional, and this has to be always kept at the back of your mind when you're selecting the lasers suitable for your dye, when you're putting the different channels imaging. So which channel has to be imaged first, which channel has to be imaged later, because if you are using a wrong sequence, you may end up bleaching your sample and you may not get enough signal for imaging purpose. So a quick look at the Jablonski diagram. I think all of you have seen this, and it's well known, but still it's so fundamentally important that whenever we excite a fluorophore with a particular wavelength and the energy associated with that wavelength, so it excites the fluorophore from the ground state, which is the resting state, to an excited state. But after going to the excited state, there are a lot of molecular vibrations in that state. So there are certain loss of light or loss of energy during the molecular vibrations. But again, it falls back to the ground state and end up giving photons, which is the signal emitted. And this signal is which is tapped for fluorescence studies. And if we plot it, we get like a excitation and emission spectra, which are plotted in form of like a Gaussian profile, and where we see the blue one is the excitation light spectra, and the green one is the emission one. So we have to collect something from this emission spectral bandwidth, and then without collecting anything from the excitation spectra. Now, typically in a fluorescence microscope, we see something like a fluorescence filter. So basic idea about a fluorescence filter is a combination of 3 filters. As you can see from the illumination part, so the light source enters. So this is the excitation filter first. There's a dichroic mirror which are positioned at 45 degree, and then we have the band filter or the emission filter which emits the light or filters the emission signal and allows the light to fall onto the detector. So if you can see, this is the schematic. So the blue filter is there, and after the blue filter, only the blue wavelength is passed through the dichroic mirror. It falls onto the sample. From sample, we may have emissions, different emissions, but the emission filter again makes sure that only the green wavelength light is passed, and finally we get images. So there are different types of filters available, and we have to understand the property of the filters also, but to a certain extent, because when we come to a confocal system, part of this fluorescence filters will not be used. But to have a basic idea, you should understand what all filters are there and see it in a wide field before coming onto a confocal system, so that you know or you have an idea what is the signal you are having in your sample. Now this is just, you can see the spectra in which the excitation peak, excitation you can see here, and this is the dichroic mirror, and this dichroic mirror in the red has been shown here, and after that this is the collection window. So anything above almost like 500 is being collected, but again the collection window, if you collect the entire stretch, we may end up getting signals from the yellow as well as orange and red regions also, but the collection has to be again specific, so something around this white spectra area is collected. Now as you can see here, from this spectra also, how we calculate, we say this is the lambda max, the center position, and the middle one, this pink line, you can see the bandwidth at half maxima. So full width half maxima has to be checked. Now why this is important? Because in a confocal microscope we have a selection availability, like we can increase or decrease our selection window as from which region we have to collect our signal. Now, if you don't understand the spectra properly, we may end up getting signals which are not actually contributed or emitted from the or your signal of interest. So as you can see here, if the proper selection is there, that the yellow line for the blue wavelength, that is the excitation wavelength, is only used for excitation, then this pink line cuts off the light. Anything above the pink line, you can see is going to the emission part, and only the emission wavelength area has to be used for the collection window. But if we do not collect the lambda maxima, that is the green peak, in that if you see you do not collect the lambda maxima area, we end up losing 80% of our signal. So although there are like ample signals in your sample, but we end up losing them unnecessarily just because our collection is not appropriate. Similarly, there is also a problem of crosstalk. If we have a collection window which overlaps with our excitation wavelength also, then the excitation energy is contributing to the emission part. So that has to be also done away with. So for a small example here, if you can see, this is for the set we have done. And if we end up collecting or signals from here, so you can see there's a lot of overlap, and based on that we are collecting signals from basically two regions, which is not supposed to happen. Rather, if we just restrict ourselves to the lambda max area of the emission spectra, we get specific signal and the unwanted signal is done away with. So imaging parameters grossly like depends. First of all, you have selected, but here the basic thing which we will be talking about is the signal to noise ratio. So as you can see, right from A to D, the same image, just because the signal to noise ratio is poor, we cannot hardly make any sense from image A and B. But as the signal to noise ratio improves, we can find a good image. Now, what is this signal to noise ratio? To understand it, this is just a representation. We have the pink or highlighted area is basically the signal which we are trying to acquire. But as we know, there are a lot of noise which are involved, noise related to the efficiency of the hardware, or noise related to the instrumentation, noise related contributed by the electronics of the system. So all this noise, we will be getting it, we'll be collecting it, and they will adversely affect the image quality. So as we can see, the signal to noise ratio, in this case, the noise has to be as minimum as possible with the best signal possible or given by your sample. So a quick example, if we see in both the images, the first and the second one, so you see in the first case, there is a decent signal to noise ratio, and that is why we can make out the difference in this also. So the beads are also visible in this area. So beads are also visible. But if the signal to noise ratio, the noise has increased slightly, the noise has overshadowed the signals which are present on this area. So only one sample is visible, or rather one bead is visible, and rest other beads are masked by the background noise. So which is also eminent from the below image, where you see a SNR of 23, and gradually if the SNR falls, so we see the background noise has increased so much that we are losing the actual signal. So to explain this, we have like small step size gradients, and as you start from 1 to 8, the noise is always there, but the gradient involved in the signal is far more prominent than the background noise. But in the other case, if the background noise is so high that we cannot differentiate between these small gradients of the signal, then the background noise will definitely mask these gradients and then it will be lost. So we will end up getting a lot of background noise and our signal will be lost in between the, in somewhere in with the noise. So in the best case scenario, if we have a very high numerical aperture objective, so 30% is the collection, objective lens which has a 95% transmission efficiency, 95% transmission efficiency, and then we have like the dichroic mirror filters which are used. They have also a transmission efficiency of 90%. The filters which are in the light path, they also should have 90% or above. And finally, the detector, which has a quantum efficiency of 95%. So if you take all this into consideration, then only we end up collecting 22% of the signals which are actually coming out from the sample. So you see, in this best case scenario also, we have such limited number of samples of photons coming out from your sample. And to enhance it, we have other hardwares which will increase the sensitivity so that out of this 22% signal which you are collecting, we can make a good image out of it. So there has been always a fight how we can improve the sensitivity of the system so that even if it is a very weak signal, even if the signal to noise ratio is very poor, but we end up getting a good image. So first thing is obviously sensitivity, the sensitivity of the detector, which is important. The light source has to be coherent. So these days in LSM 980 also, there are solid state laser lines are being used. They are coherent sources of light, and that will increase the sensitivity. And with that, obviously, for dynamic experiments where there are a lot of like you have to have a number of frames has to be high over a time lapse experiment, and there is a movement happening like a diffusion studies. So in that, speed is also very important. So sensitivity along with speed, and not to forget the depth of the sample. So if you have a thicker sample and you want to go deep into the tissue and you want to image it as good as we do it on the surface, then that has to be also taken into consideration. So this is image you can see here. This is a Drosophila image, and you can see this looks like a donut when it is properly resolved, but when it's properly not resolved, we see something like a blur, like a big blob out of it. So resolution is another important thing which has to be definitely there when you are taking care of the imaging parameters. Now, when we have all these things at the back of our mind before taking image in a confocal system, we should also have an idea at what is my sample size and what type of sample I'm using, what should be the best technique to address my sample and then get the best image out of it. But again, the basic question is, why do we need a confocal system? We can do this with a wide field fluorescence also, but why do we need to have a confocal system? Because if you see, this is the sample, and if you can consider the blue central area as a nucleus and the red spots, they are like mitochondria. So it's like visualizing moon from the top of your terrace, and you see stars, clouds, moon, and you have an apparent impression that they are all lying in the same plane. Basically, it is not true, as they are all distant from each other. And so to understand that, we need to have a confocal system. Okay, so if we compare with a wide field and a confocal laser scanning microscope, the first thing is obviously the light source, which has been replaced from a mercury light source to a laser light source here to have more coherent light source falling onto the sample. And then the other most important thing is obviously the pinhole, which is placed at the conjugal focal plane, which will chop out the out of focus light and only allow the in focus signals of light to fall onto the detector. And finally, instead of a camera, we have a PMT photomultiplier tube which detects the signal. So this is a point scanning detection. Now, as you can see, the light illumination in a wide field is is the entire sample in that Z is being illuminated, and we have the background as well as the plane of focus. So when we collect the signal, we also end up collecting background signal, which is out of focus signals. But when we compare in a confocal system, we see is like a Z stack, which is done. Optical sections can be possible, and that is by the pinhole virtue. So if you see the light falling, this is the plane of focus, the yellow dotted line. I'm able to collect the plane of focus, and only the light is allowed to pass to the detector, right? But at the same time, we are also illuminating the out of plane above and below of the sample. So we'll also get emission coming from above the sample or above the plane of focus, and also below the plane of focus. But the thing is, the signals coming from out of focus plane will not meet at the conjugal focal plane where the pinhole aperture is being kept. So this pinhole aperture is basically kept with a one array unit, and this will allow only the in focus signal to pass and reach to the detector. So if you see in a wide field, we get something image of a pollen grain like this, which is just the surface impression, and there are no details information having. But as in with the virtue of a pinhole and the confocal system, we can virtually slice the sample and can go deep inside and find the exine entine differences without even touching the sample. So this is just the Z stack of Drosophila I, and as you can see, the different Z stack images which are taken at different Z positions, and when they are reconstructed in a 3D, we get a complete three-dimensional image of a Drosophila I. So that is the power of a confocal microscope. Now, if you see the XY galvanometer, there's a galvanometer is basically a scanner which scans the sample in a XY direction. So in the XY direction, it will change in the XY plane, and it will cover the entire XY plane of the sample from the plane of focus, and we get spectras from the particular line. So one line, if it scans, we get an intensity, and based on the fluorophores, which these intensities are even converted into a 2D form to give you a final image. Now this is the ZEISS LSM 980. I'll quickly show the light path. So in this, we have the white line which is coming from the solid state laser lines, and then it is coming onto your sample through the objective. Once it reaches to the objective, the emission signal comes back. So emission signal comes back, and if you can see in the fast linear scan mirror, so they're getting descanned, and by descanning it is directed to the different twin gate dichroic mirrors, and which are again there is a recycling loop to compensate for the loss of the light which may happen due to reflection of the certain mirrors, and then finally it is reflected back to the detectors. There are two flanking PMT detectors, and in the center there is a gas detector. So the flanking PMT detectors are efficient in the like near to UV or in the blue region, and also in the like 740 nanometer and above, and the gas detector is high sensitive, and which works well within the range of 440 to nearly 650 nanometer. So in a wide field, basically a camera chip is there, and the entire camera chip is illuminated at the same time. The pixelation is fixed. We do not have any control over that. So the entire like the chip is illuminated, and we acquire the signal. But when we come to a confocal, it's a point scanning detection. So pixel to pixel information is being collected, and then based on the number of pixelations we have selected, the PMTs keep on converting from the photons, number of photons, to electronic signals, and they are converted into form of an image. Now we have the laser light spot, which is the minimum light spot which is achievable is 256 nanometer, and based on this, if we keep on scanning, we can see the pixel size and the minimum spot of the laser light is correlated, because if I have a very large pixel size, then the spot will fit into it, as in this case. If you see the pixel size and the spot of the light which is achievable, it will keep on scanning, and in a minimum time it will complete the entire frame and give the best image possible. So 512 by 512 is the pixelation which is conventionally kept for a confocal microscope, and that was like serving the purpose good enough for quite a long, until unless people have looked for more resolutions and they have started to playing around and pushing the boundaries to 1K by 1K, 2K by 2K. But one thing has to be understood: the spot of this light, illumination light source, is fixed. So we cannot reduce the spot of this illumination light less than 256 nanometer. Now, if we reduce the pixel size to very small size, what will happen? The spot or the illumination spot, which you can see here in the blue color, it will spend more time to cover the same field of view which was covered in the previous. So the field of view has not changed, but the pixelations, because the pixel size has been reduced, the number of pixels have increased, the time taken for the spot of the light to capture the area to cover the entire field of view has increased, and it may end up bleaching your sample. So there has to be an optimal ratio till what should be your pixel size compared to your like what is the tightest spot, 256 nanometer, as I told you. You have to always keep in mind, and based on that, conventionally 512 by 512 is the pixelation which is considered. We can definitely increase the resolution a bit if you go for 1K by 1K, and at times to 2K by 2K. So if you see here, if we compare again the Z resolution in a wide field, so the wide field image, you see there's a lot of blur there, but in case of a confocal system, you have much in focus and there's a lot of background which has been done away with in the confocal system. Now, there's one more important aspect which has to be always understood, because the pinhole aperture of the confocal will decide the confocality of the image. If the pinhole aperture is open, it will behave like a normal wide field. So what should be the pinhole aperture, and how we can determine the size of it or diameter of it? Now, as you can see here, the laser line is being going and putting, and by passing through this pinhole, it is reaching to the PMT here. Now, the mechanical pinhole definitely rejects a lot of photons which are out of focus, and that is why the collection window is again reduced. So we are getting a very, very less number of photons in the collection window. But the collection or the diameter of this pinhole will also determine the section, the Z section thickness of the sample, or what is the best Z section resolution which we can achieve. So it is done usually on the one array unit, and is a like as you can see here, the Gaussian profile again. So if it is a perfectly the smallest dot of light which is falling onto the sample, so a perfect Gaussian profile will give you the point spread function, and if you can cover the perfect point spread function, it will give you the one array unit distance, and based on this one array unit, the pinhole diameter is adjusted so that you get the best confocal image. Now, this will also improve the optical sectioning image. So in the Z plane, whatever optical section we have been doing it, but there are other parameters which has to be always kept in mind: the numerical aperture of the objectives you are using, the wavelength of light which is being used, and definitely the pinhole diameter again. So all these three works in combination to give you the perfect confocal image, and the limit or the resolution limit of the system can be achieved easily with the best lenses. So there are like multiple detectors are there, and how they are being guided. So we see the laser light falling onto the sample and just passing through the pinhole. There is again the filter is there, a long pass filter. So anything above 505 nanometer is being collected to the PMT. This is for one channel. But if you have another like two dyes at the same time, so two color simultaneous imaging if you do, so what will happen? We have to put another beam splitter or a secondary dichroic mirror, which will have which will reflect something from like 505 to 540. So anything 540 or 550 nanometer above will be passed straight, and then it will be collected on the second detector. So simultaneously we can detect using two detectors, and two color simultaneous imaging is possible in this manner. Now, in a fluorescence, there is always the number of photons which you are collecting is very less. So in the best case scenario, we saw something around 25% approximately photons we are collecting. Now we have to create an image out of 25%, which is a tough job, which is done by the detector. Now this is not a normal detector. This is like a photomultiplier tube. The photomultiplier tube's role is to amplify. So you can see here the blue diodes are placed, which are positioned in an increased potential difference. And when a single incident photon comes and strikes to it, it keeps on multiplying and goes to different, like higher potential gradient, and the number of photons is getting amplified. So finally, at the receiving end, when we get the number of signals, that the intensity of signals is multiplied, and thus it helps to give you a better image. With advance in technologies, the detector has also improved. So in LSM 980, not only the normal conventional PMT multi-alkali photomultiplier tube is given, but along with it, a higher sensitive gas detector, which is basically from gallium arsenide phosphate. And this is a semiconductor material which is used or considered very ideal for converting photons into electric signals. As you can see from the quantum efficiency plot, compared to the array PMTs, compared with the cooled PMTs also, it is almost like 45% quantum efficiency is achieved. But from a limited wavelength range, that is why a flanking PMTs are also given along with combination with the gas detectors. Now, a simple image comparison, if it's done for PMT as well as a gas detector, so both the detectors have acquired the same image, and if you can see the spectra here, the spectra, you can see the background noise in this red signal. You see this red arrow, the same line has been plotted here. You see a lot of background noise. So this background noise has definitely dampened the signal intensity, and the signal to noise ratio has deteriorated considerably. But compared to a gas detector where the quantum efficiency is higher, the signals are quite prominent, and the background noise, as you can see from the image itself, it's quite black, whereas in the previous one it was all dotted. Now, we can use gas detector and how we can capitalize the potential of a gas detector for a better imaging purpose. Definitely we can have better image quality with the gas detector, higher sensitivity, which is like improved SNR. It's not good in SNR, but also it is faster, so it scans in a faster time compared to normal PMTs. And for longer imaging, so if you have live cell imaging, which is a time lapse experiment going through overnight, then there is also photo bleaching issue associated with it, and even the laser power, if it is high, they may have a phototoxicity induced in your live cell. So to do away with that, even if you are using a gas detector with a very less nominal laser power, you can record it for a longer time without even bleaching your sample, which you can see here in these two cases. Even in these two comparisons, the same sample which is acquired from a gas detector over a period of time, you see the signal intensity is almost constant, but with the PMT, you see the B, there is a loss. Now, how this laser scans it from point to point? So there are basically the conventional method is unidirectional. So it goes from X0 to X512, comes back, there's a beam blanking flyback, comes to the homing position, and there is a one shift in the Y scale, and again X0 to X512 is the movement. So as you can see, the readout is only in one direction, and as it readouts, it collects information in that particular frame. There are also methods, if the sample bleaches out very fast, you can have the same activity or acquisition done in a bidirectional method where there will be no beam blanking flyback at the end of 512, like X512. Rather, it will be in a zigzag fashion. So once it completes in one line, it will in the same direction, it will move in one Y, and then it will keep recording on the back to the X plane again. So this is like a zigzag. The half of the time is saved. So but the trade-off is there should be XY pixel alignment, and it has to be like always checked that your XY pixel alignment is perfect, because it is doing at a faster rate. Now, so the speed and averaging, we can also, if the signal to noise ratio is poor, your signal intensity is very less, still you want to have a good image, then you can always have averaging out. So as you can see here from the acquisition, scan mode is usually done in a frame. The entire frame of one channel is being done. 512 or 512 is the like conventional pixelation which has been followed for confocal microscopy. And in the red highlighted box, as you can see here, the pixel dwell time. So this is the time, as you can see, 1.6 microsecond is the time taken for the detection of per pixel. So if you increase the pixel dwell time, the more and more photons can be collected. But yes, the speed will be slow, sensitivity will improve definitely, but you can also have a risk of bleaching your sample. So what should be the pixel dwell time? The entire scan time also will be affected because of it, and based on your signal intensity which you are getting from your sample, this dwell time has to be decided. If your signal to noise ratio is very good, your dwell time can be reduced to, and you can acquire the same field of view in a very less time. And there is also like averaging out. You can average it out to like the same acquisition for one line can be averaged out to twice, or and the average intensity will be plotted, but it will again increase the time. The other thing is, like whenever we are increasing the signal to noise ratio in case of an averaging will also be affected, because if you can see here, if n is the signal number of photons you are getting, this will also contribute to the noise, background noise. And so in ideal condition, your signal intensity should be higher. Signal should be very high compared to the noise you are getting. And that is why if you increase the dwell time, you may end up getting good signal, but your short noise vibrations may also effectively or contribute to your signal. So which will also in a way decrease your signal to noise ratio. Now, if you can compare these two images, the first one is a good image where the background intensity is blank. So there is no background noise coming. In the second one is noisy, and that is why you can see a lot of dots, pixelations. Now, once we have done it, we have to also understand that confocal is used to quantitate intensities. So this intensity quantitation or comparison between two samples has to be done in a very effective manner, and there has to be a proper scaling to it. So if you see in a 8-bit 2 to the power 8, 256 is the gray scale. So as 0 to white, you see red and blue. So when we see the range indicator in a sample, it should be like there should not be any red, and the signal should be in grayscale, and the background should also do not have anything in blue, it should be in gray. So it should be within the scalable range of within 255 gray values. Now, if we have this particular spectra, which looks good, that the full dynamic range is being used, and we see the maximum intensity of the signal is within the range of 256, and the 0 is also is there. But if we have a saturation where you can see there is loss of information because the intensity has overshoot the maximum scale here. So when you see a range indicator, the red like this, it is the detectors have been saturated, and this image will not be used or cannot be used for comparison quantitatively. Then obviously the detectors and the laser power. So what lasers we are using, we have choice of lasers. In this 7 lasers are there which covers the entire visible spectrum, starting from 405 to 633, and these are all solid state lasers. The laser power has to be used to the minimum. When you are using a high sensitive detector, definitely the laser power is bare minimum, like 1% or even less than that. And the gain, the digital offset and the digital gain, these are all the controls for the detector. If you increase the voltage of the detector or the master gain of the detector, the potential difference in the detector increases so high that it will try to collect more number of photons, and in the process it will also attract background noise. So the digital gain should be half of the entire dynamic range, which is 620 roughly, and the entire is 1250. So half of that is is considered very ideal. The digital offset is the threshold intensity level, which will deduct the background intensity from the main signal. So it will automatically put a threshold limit and the background noise will be deducted from it. And the digital gain, in case we do not have a good signal, then we want to amplify the signal, but in the process the noise will also get amplified. So in the digital gain should be the last resort to look for. The information given, the Z resolution, I think like Dr. Tony has extensively talked on this. So I will just pass on with this, but what you have to understand what objective we are using. So the best objective is with the highest numerical aperture, which is a 63x 1.4 numerical aperture. And you can see the information on the top of the objective diagram, 1.4 oil. So this objective will be used to use the maximum resolution capacity of the system. But in addition, we can also have a magnification created for your image with the help of zoom. Now zoom here in a confocal system, as we have the virtue of selecting the pixelations of imaging by changing the pixels, number of pixels involved in imaging, we have the leverage of zooming in, zooming out without compromising on the resolution. So in the first image, you can see from 512 by 512 to 1K by 1K also, if you are increasing the pixelations, the scanning is done for the same field of view. So there is no change in the field of view, the same image size is being done. But if we scan from like zoom 1 to zoom 2, the number of pixels doesn't change, but the field of view decreases. So with like a magnified image for a very specific region of the entire image, we can also zoom in and take it with the same resolution which has been acquired for the larger one without compromising on the resolution. Now, when you're talking of resolution of a z-axis is also very important. In the z-axis, once we set the entire thickness of the sample, the software will tell the optimal thickness. The optimal thickness is always based on the Nyquist theorem. The Nyquist theorem will always say that sampling frequency, whatever is done, or basically the slice thickness, it should always be double of the information frequency or the resolution you are looking for. So based on this, if you select the software has the capacity to tell the smallest one, which is highlighted in the red box. And if we say click on that, it will give you a resolution of, like, as you can see here, 390 nanometer is the resolution which is achievable. And in this image, the slice intervals also can be decided. So based on the resolution, we can identify how many slices we can actually image with that interval for a range of 20.12 micron. Now, what is the advantage when we have, so this is an example of an adult mouse brain, and the neurons, if you see, you can see the clarity over a z-axis, and we can reconstruct it in the form of a 3D structure. So settings, there are plenty, but what we have to know, or what we should have at the back of our mind when we are looking for a confocal imaging, or when you are coming for coming to a confocal facility. So as sample design, how you are going to prepare your sample, what type of vessel you are using, a glass slide or a cover slip bottom glass Petri plates you're using, 35 mm or a 60 mm you're using, or lab-tech chambers you're using. So all these glass vessels are allowed. The thickness, if you are going for 60x or even 40x and above, in that case, the thickness should be less than 1 point or even less than 1 mm. The cover slip or the glass slide you're using, it should be of 1.5 grade. So there are no diffractions within the glass slide. The dye should be specifically stained. It should be in good quality so that you don't have non-specific staining that will be adding up to the background. And the sample preparation should be as good as possible. Apart from that, the objective, so what objective you are looking for? The objective is obviously, if you go for higher objectives, the numerical aperture is higher, the resolution improves. But yes, then if you want to focus onto a single cell, then there are always options with the 40x or 63x also. You can zoom in to a single cell imaging with a very fast scanning. using optimally the laser power, managing the settings for the detector so that you get an optimal signal, and finally using the best detector in case you have a low signal to noise ratio. And also the pixelations, like from 512 by 512, you can push your boundaries to 1K by 1K, whichever is optimal. And then if it is a simultaneous, like if you have a colocalization studies where two proteins are being secreted at the same time, you want to have a colocalization experiment done, then you have to do it at the simultaneous. Or in other case, in order to avoid crosstalk, photo bleaching, or phototoxicity, and if you want to like avoid all those things, then a sequential will be much smarter or a safer option. So as you can see here, there are a lot of dyes with the different spectrals. They do overlap a lot. So in this case, it is always good to have a sequential one after the another and then image them sequentially. So if you can see like dynamic processes also, so when you can see a photo conversion experiment, where there is like a change over a period of time. These are like time-lapse experiment, and in both the cases, like also in photo recovery, like a diffusion experiment where you want to bleach an area and after that you want to see how fast it is recovering. This all experiments are possible in a confocal system. Again, these are all correlative studies. So the dynamic properties of the labeled proteins which you are using, that matters a lot. What is the excitation? So you understand the excitation volume. You are exciting the entire volume, and how many frames you want to do it to cover your interest of region, how fast you do it, so that you have to keep in mind the rate of change in the dynamicity of the experiment. So keeping that in mind, how fast you need to push your system so that you acquire all the activities happening in your experiment. And definitely if there are any interactions happening with your interventions you are giving, maybe a drug or maybe any chemical interventions or whatever interventions you are giving at the middle of your experiment, you want to have a pre and the post intervention images, you can definitely acquire them. So the birth of live cell imaging, this is a short video. It's back to 1945, somewhere like a microcinematograph, where we can see the first time a cell division has been, the active process has been videographed in the form of like a movie. And with years of advancement, these things have been much more improved. The dyes have been much more specific, and as you can see in the kidney epithelial cells, the cells are dividing, whereas in the microtubules changes in Arabidopsis thaliana. So all these specific activities can be imaged with a higher frame rate, and we can have much in-depth information over a period of time. So the time resolution, the temporal resolution has also improved, and we are able to achieve images of this quality. And this is the last one because Arrivis has powered the 3D visualization of the Zen software, which you are using with the LSM 980. With the Arrivis software, the 3D is a cutting edge technology. As you can see here, we can have a lot of 3D images. We can do a Z-stack, reconstruct it into beautiful 3D images, and then can have beautiful videos which will give much more meaningful information to your studies. Thank you.